So let me tell you the post-transcriptional modification, what actually is. I will cover you everything regarding this topic, okay? So first, from your template DNA, at first the RNA is produced, okay? This process actually called the transcription. It is the first step of gene expression. That means from your template DNA, mRNA is produced. This initial mRNA basically called the primary transcript. We also called it heterogeneous mRNA. Okay, so this heterogeneous mRNA is basically non-functional. It needs some modification to become functional one and to leave the nucleus. Okay, so first modification basically the 5' capping and 3' poly A tail. So 5' capping means modified guanine nucleotide basically added on the 5' end of your heterogeneous mRNA. It basically 7-methyl guanosine. Okay, some modified nucleotide as you can hearing. Okay, and then tailing. The 3' tailing a string of 200 adenine nucleotide basically added one by one. Okay, and then your mRNA become 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 from heterogeneous mRNA to pre-mRNA. Okay, heterogeneous mRNA and primary transcript both are same, both term is same, and then whenever the pre-mRNA is produced, it need another modification that called splicing. Splicing means removing of introns and joining of exons. So whenever the introns are removed, that means the non-coding part are removed, they are non-functional. Okay, so non-coding part are removed from your pre-mRNA, and then the coding part are joined, and coding part, we know they code for specific amino acid, or we can say a specific protein entirely mRNA basically. Okay, I'm talking about. So whenever they join together, spliceosome, here is complex, and spliceosome there is a long process, but in our cell it occurs in a very short time, but this process is long. Okay, then whenever the splicing occurs, your pre-mRNA become the mature mRNA. Now your mature mRNA is ready to leave the nucleus. From your nucleus, it will go towards cytoplasm, basically towards the ribosome, and the ribosome will translate the code from your mature mRNA, that basically carrying the message from your template DNA. Okay, then the protein will produced, and then your protein needs some modification, that called post translational modification. Okay, so like the phosphorylation, the glycosylation, methylation, the types of modification basically need for a protein to become functional one. Okay, so why we need post translational modification? To make a protein functional, because initial stage of your protein, it's a polypeptide chain and it's not functional one. Okay, to become functional, it needs to undergo its three-dimensional shape, and its three-dimensional shape or 3D shape is functional shape. When it goes some modification by the hydrophobic region, or the non-covalent bond, okay, many interaction are occur. Okay, then it will go its native conformation, and that conformation is functional for a specific protein. Okay, so that's it for today. Thanks for hearing.